Hello friends, welcome to my channel Dr. Sam's Biology. In this video, we will discuss about highly repeated DNA sequences. So what are these highly repeated DNA sequences? These are the sequences of nucleotide or sequences of DNA that are repeated over and over without interruption. Means they show tandem arrangement. Means they repeat one after another in continuous manner without interruption. So this is example of highly repeated DNA sequence. In this sequence, you can see that this is one sequence. AT, AGT, CGCT, and then it is repeated again. Then it is again. So it repeats over and over without interruption. Means one after another. There is no interruption between these sequences. So this type of arrangement is called tandem arrangement. When one DNA sequence is repeated just the end of this sequence, just after the end. So this is repeated over and over. So these are DNA sequences which are repeated over and over without interruption in the DNA or in our genome. So these type of sequences are called highly repeated DNA sequences. They fall in three categories. First is satellite DNA. These are short DNA sequences, short nucleotide sequences of 10 to 1000 base pair. So these are short sequences of 10 to 1000 base pair that are repeated over and over in tandem manner without interruption. These sequences are clustered together and repeated over and over. So this is an example of satellite DNA sequence. In this example, you can see that 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So this is a sequence of 10 nucleotides. Then it is repeated just after its end. Then it's repeated. So it repeats over and over without interruption. And these are 10 to 100 base pairs. These sequences are called satellite because the DNA fragment of these sequences form distinct band. So when we perform centrifugation in centrifugation tube, they will form specific bands like this. That's why they are called satellite DNA. And these sequences are found near centromere and telomere of chromosome. So this is the chromosome. We know that this part is centromere. This is centromere. So these sequences are present around centromere, and this end of chromosome it is telomere. So these sequences are found near centromere and telomere. These are called satellite DNA. Next is mini satellite. As its name is indicating, these are repeated sequences which are shorter than satellite DNA. We know that satellite DNA sequences are 10 to 1000 base pair. But in case of mini satellite, they are around, or you can say up to 15 base pair. So these are up to 50. These are not more than 15 base pair. They are a sequence of 15 base pair. It will repeat 15, 15. It will repeat over and over again in tandem manner. So the difference between satellite and mini satellite is that they are longer sequences. These are shorter sequences. These mini satellite also found near centromere and telomere of chromosome. Our third category is microsatellite. As its name is indicating, it is more shorter. It is shorter than this mini satellite. So these are shortest repetitive sequence. Largest repetitive sequence, satellite. Shorter than satellite is mini satellite. And shortest is microsatellite. These are around 2 to 5 base pair long. These are scattered throughout DNA length in chromosome. These are not present in, or you can say present at specific places like centromere or telomere, as in case of satellite DNA or mini satellite. These are scattered throughout chromosome. If it is a chromosome, it has DNA. So these sequences are scattered throughout this, or throughout the portion of chromosome. Their position and number of copies varies person to person. Position means where they are present on a chromosome, and their number. In some person, their number may be 500, in another it may be 500, some other number. So it varies person to person. So this is an example of microsatellite. So here we have three nucleotide sequences A, T, G, and it is repeated over and over tandem. So this is highly repeated DNA sequences. They are repeated over and over again in tandem manner in whole genome, or you can say in a whole chromosome or whole DNA. They fall in three categories. Satellite, these are longer sequences, 10 to 1000 base pair. They are repeated over and over again. Mini satellite, shorter than satellite, they are up to 15 base pair. Microsatellite, they are shortest repetitive sequences. They are around 2 to 5 base pair long. So this is highly repeated DNA sequences.